All right, good morning. Today we're going to go over how to interpret your DNA sequencing results from a StrainSEEK report. There's several different ways that we sequence for clients. StrainSEEK is one of the more popular ones. This sequences about 3.2 million bases in the cannabis genome. This is different than the whole genome sequencing service we have, which sequences every base, over 800 million. But the StrainSEEK tool is a good, cheap way to get in and get some surveying done. A lot of people are using this for intellectual property protection. If you actually look into the patent office, you'll see many plant patents out there that have names that are actually in Cannapedia. So it's probably the most popular tool for cataloging novel strains, but it's also very helpful for breeding because there's lots of information in these reports that we're going to go through that can help you with your breeding efforts. So let's go through some of the tools here. When you do sequence with us, the SureSelect system from Agilent is being used in this case. We also have some panels that we're developing with Arbor, and you'll see those shortly. But this sequences 3.2 million bases in the plant, and it does it with a targeted hybridization capture that can collect any region of the genome and sequence it. And in this case, we have selected around 25,000 SNPs that can be detected with the sequencing panel. But we've also tiled some key genes. Over 926 genes have variants in them that you can track as tag SNPs, but we also target CBD synthase, THC synthase, and cannabichromine synthase very heavily so you can get full gene coverage over those genes. This means you can look for those genes, mutations in those genes, or if those genes are deleted with this tool. So here's an example of a sample known as Blue Dream. Blue Dream is quite common. And what you can tell by this report over here on the right, this is a violin plot that tells you how similar your strain is to everything else in the database. So strains that have a lot of commonality to other plants in the database have a fairly bottom-heavy violin plot. If it's a very rare plot, you will see this plot change. If it's a very rare strain, it'll become more top heavy. You'll see more of the strains in the database being very distant from your strain. So this is basically a density plot of the strains that are in the database, and the lower this gets, the closer it is to your strain. So as this becomes more bottom heavy, you have a more common strain. As it becomes more top heavy, you have a more rare strain. Some of the descriptions are right down here and will pop up for you to describe what these mean. Now, we collect an RSP number. That's a number that is unique in the database, and it's unique to your cultivar. And we often collect other bits of information that people provide with the sample submission. Sometimes they will supply us with cannabinoid terpene information, and you're going to see that in a couple of reports going forward. All right, so the first thing that we tend to look at here is the heterozygosity plot over here. This tells you how different you are from the reference genome. The reference genome that we're using here is Jamaican line. We have this published in a preprint that you can view here. This preprint here has 42 other genomes aligned to the Jamaican line genome. And these are whole genome sequences. We also have some RNA sequence in here, some IsoSeq, which is RNA sequencing, and also some methylation data that's in this paper. And all of this data is also in a genome browser known as Koji, which we're going to touch on in a little bit. Okay, so that's the reference that we're comparing everything to. And your heterozygosity is a number of variants that differ from that reference, and we calculate those and quantitate how heterozygous your plant is. When you're breeding for plants to become more stable and produce more stable seeds, you want to see this green line move more in this direction, so it is less heterozygous. And that's what a lot of people do, is they back cross to watch this thing move in that direction. Okay, there's a little pop-up here that describes the heterozygosity as well, if you need that. And then the other most important thing we're going to look at is the coverage over particular genes. Okay, this is sequencing read coverage over what we've identified as the BT-BD allele. Okay, the BT-BD allele, Etienne DeMeyer published, they theorized that there are regions in the genome that dictate whether the plant's going to make THC or CBD. Okay, now so while Etienne DeMeyer theorized that there were markers in the genome that might track with this, they did not actually distill down the genetic locations that might drive this. And so we spent a lot of time sequencing many genomes to try and sort this problem out. We've got a couple preprints that describe it, but lo and behold, there's a couple ways that this BTBD allele actually operates. There's not a it's almost like convergent evolution. There's not one solution to the problem, but the vast majority of the genotypes we see out there are related to deletions of entire genes. Okay. So a couple other markers have been described in the past, known as the BTBD allele. They don't actually reside in the THC synthase or CBD synthase genes. They're close to it, but as a result of their distance from the genes, they may not be 100% predictive. So what we've switched to is actually monitoring the presence and absence of these DNA sequences, the genes themselves, in the genome. Now that we have these really great references, we can count how many cannabinoid synthase genes are present in your genome. So that's what these plots are depicting here, is they're depicting your Illumina sequence coverage over the CBD synthase gene. That's on contig 1772 in the Jamaican line reference. And then it's also looking at the DNA sequence coverage over contig 7741 in the Jamaican line reference, which has THC synthase in it. It also has a neighboring gene that looks a lot like CBD synthase but is broken. Okay, this is what we call inactive CBD synthase, all right? This happens to be in very close proximity to this. And so we're only tracking this CBD synthase as a confirmation of this marker, and to also try and track how large this deletion is. What we tend to see in most genomes is full deletions of the CBD synthase gene or full deletions of the THC synthase gene, and it usually embodies this neighboring cannabinoid synthase gene as well. All right, so this is a little bit redundant. This blue marker should almost always correlate with this green one. If it doesn't, you have something unique where the deletion might be a partial deletion and it's taking out just a piece of this gene, but it's probably chemotypically irrelevant. This is something that we're just tracking to get a better size of the deletion of that gene. Okay. So this can be very helpful in deciding whether you have type 1, 2, or 3 plants. Now, this is a complicated loci. There are a couple of things that can go on. I said convergent evolution before. What does that mean? Well, that means there's a couple different ways that you can kill THC synthase. You can either delete the entire gene, or you can have point mutations in the gene that knock out its function. The gene is still there, the RNA is probably still made. However, the protein that gets made from that gene does not fold CBG into an appropriate cannabinoid, and so it's the gene is there, but it's not functional. That's a very, very rare genotype. Probably only 3 or 4 in the database have point mutations like this, and we have probably over 1,000 in there now. So this is that's an exception, and it's also a little bit hard to predict because sometimes those point mutations aren't always in the same place. They can often be novel. We see a lot of activity at the 333 amino acid location, and that has been published in one of our preprints, P333R. And there's a couple other places we've seen hit as well that are right now in private reports, and we can't speak to what those are, but they're nevertheless, when we have seen variants in this gene, it's very rare they're the same. They always seem to be somewhat private and unique to people's cultivars, which means they have something novel, and they usually end up reporting that to an intellectual property attorney or looking into plant patents when they find something like that. So, all right, so anyway, we're going to go through a couple of these plants to show you how these graphs change from type 1, 2, 3, and 4 plants, okay? This is Blue Dream. You can see all of its relatives here that are near clonal distances apart, and some other ones that are a little bit further away. Now, this chart down here has a clone distance and a sibling distance and a little fuzzy area here. And the reason this is fuzzy is that you can have some inbred plants that when you cross them, they come out with siblings that blur this line a little bit. Most siblings are quite distant if the parents are distant, but if the parents are very similar, their siblings are going to come out a little bit tighter. So if you actually have an inbred line, you may see some of the siblings get closer and closer to this line. Likewise, self plants tend to bend the rules a little bit more this direction as well. So this isn't by any means a hard and fast definition of a clone and sibling. It is a genetic distance score, and the better and better you get at inbreeding, the more you're going to move from sibling to clone. Okay, so that's Blue Dream. That's clearly a type 1 plant. It's got a THC gene. It doesn't have a CBD gene. All right, let's look at the original Jamaican lion plant. All right, this is not the Jamaican lion that we did the huge sequencing project on. After we had sequenced the Jamaican lion reference and put it out there suggesting it's a type 2 plant that might predate the biotech patent, the breeder of it came forward and gave us the original from 2011, and we sequenced that as well. Okay, now since this was the original from 2011, it was what is that, 7 or 8 years predating the one that we sequenced. So of course it's not identical, it's not a clone, but it is got a very low heterozygosity because we are comparing Jeremy Menard's original Jamaican line from 2011 to the 2019 descendants, and of course there's going to be some variation after breeding. And so the heterozygosity is way low on the charts. And you can see in here, a couple of these Jamaican lines descendants are getting very close to the clonal distance line because they are closely related to the original genetics. You'll also notice we have an active CBD gene and an active THC synthase gene. All right, this means it's a type 2 plant. Again, this one's coming along for the ride. Let's take a look at Queen Jesus. Queen Jesus is an interesting one that we've sequenced. It has a chemotype in the database, and you'll notice it doesn't make any cannabichromene. And what we've also noticed is that we need to add another plot here to look at the cannabichromene genes, because we do in fact cover those in your StrainSEEK report. And you would find that in this VCF file. If you went and looked for cannabichromene, there's 8 of those genes on contig 756. And you can chart the coverage over those genes to determine whether you have a cannabichromene deletion and get a plot just like this for cannabichromene. Why do we care about cannabichromene? Well, here's a plant that makes CBD, doesn't have a THC synthase gene, makes a little THC. All right, why is that? Well, there's a couple theories. There's a good paper I'll point you to from Zerbel that goes over this, but you can, CBD synthase and THC synthase, and for that matter, CBD synthase, they are not believed to be pure enzymes in that they 100% make only one cannabinoid. They're believed to be a little promiscuous, in that CBD synthase, from most studies, demonstrate a 23-to-1 ratio of CBD to THC synthesis. That's in plants, okay? Now, plants are very complicated to measure this ratio in because plants have all like 90 other genes that look like cannabinoid synthesis that might be contributing to this problem. However, most of those look a little broken and distant, so the ones that we really keep our eye on are 3 genes in particular: THC synthase, CBD synthase, and cannabichromene synthase. These are all within 90% sequence identity of each other and are likely the culprits for making the compounds that we're dealing with. So you have a CBD synthesis gene here. It makes lots of CBD, but this is probably one of those 23-to-1 ratios where it's leaking some THC synthase. If you take CBD synthase and clone it into yeast and start mucking around with the pH, you'll see that it can make varying amounts of THC synthase. So when the gene is taken in isolation out of the plant and put into a model organism, and you ask it to give it precursor and you ask it to make cannabinoids, it doesn't make purely CBD synthase. It leaks a little THC synthase. The same thing is true with THC synthase. If you look in type 1 plants that don't have any CBD synthase, there's still a little bit of residual CBD coming through, okay? That's likely THC synthase being promiscuous and making a little bit of CBD, although this one seems to be more promiscuous of an enzyme than this one. We tend to see a larger ratio of THC to CBD with type 1 plants than we see with CBD to THC with type 3 plants, okay? So this here's a type 3 plant. The way we know this is there's sequence coverage over the CBD gene. There isn't any over the THC synthase gene, and its neighboring gene is gone too. All right, let's take a look at a type 4 plant. What does that look like? Well, type 4 plants, like Xanthica 27, don't have any of the genes. So these are going to stop and make the precursor, CBG. Now, we don't have the chemotype for this, but if you look at some chemotypes online for type 4 plants, you'll see, even though they don't have a THC gene and they don't have a CBD gene, they still make a little bit of THC. And that is largely believed right now, although it is hypothesis, is largely believed to be coming from cannabichromene. Cannabichromene is the next closest relative to these 2 other synthase genes, which is why we're soon going to put a little cannabichromene synthase gene coverage map over here so you can chart that as well. The current StrainSEEK panel does in fact sequence cannabichromene synthases. There's 8 of those genes in the genome, so we look at the coverage at all 8. We do cover all 8, and you can find that in this VCF file here. If you wanted to comb through the data yourself, the best thing you could do, at least if you're on a Mac, is hit the control button on your keyboard and then click on this, and you'll get a window like this to open. Go to copy link. When you go to copy that link, you can then download this to your Mac. Now the reason we're going to go and do some terminal is if you just click on that near a Mac, it's going to try and download a VCF file, and some computers will auto open that file as if it is a mail contact file. And as a result of that, it will probably corrupt the file. And you don't want to deal with that. So you can do it the old school way, which is open up a terminal like this, type in a command called curl -o for output, copy and paste the link that you just copied, and down comes your file. Actually, I want to see what we have. I think I already have it downloaded here, so it's giving me a bit of an error. But there's a VCF file right there. So let's unzip that VCF file, and we can start to look at what that file looks like. And don't want to open it when it's zipped because it'll be binary and be a big mess. But if you were to more this file, you're going to see a pretty big header. All right, this tells you we embed all this stuff in the VCF file because this is the file that we hash into the blockchain. So when you do that, you want to have the people who sent it in, any of the limonene and terpene information, any of the CBD information, the chemotype we want to embed in the file. So the owner, the chemotype, and the genetics end up in a hash in a blockchain. And then after this header is done, it's going to give you the length of every single config that's in the assembly, which is a little boring. But you can skip this header if you just grep the file with a -v command. It will skip over anything that has these pound commands. I'll show you how to do that in a second. So here what you're seeing is a list of every config that's been sequenced, the coordinates, and the base and how it differs, whether it's low quality or a passing quality, and then the genotype. This is the important thing over here. This genotype right there is 00. That means it is heterozygous. When you see things that are 01, it means, that means it's homozygous. When you see things that are 01, it means it is heterozygous, okay? When you see dots like this, it didn't have enough coverage to make a call, so there's a low quality, or it is, yeah, right there, those are all low quality, those are passes. In here are different bits of information in terms of how many reads are covering it and quality scores and whatnot. You can look about what all these little details mean if you just look up VCF files on the internet. It will explain to you the file format and all the other information that's encoded in these comma-separated forms over here. But this is really the critical key because this has your genotype right there. That's the thing you want to look for. This is the coordinate and where. So inside this file, you're going to see lots of different insertions and deletions and genotypes. And it's a very big file because we are covering 3.2 million bases. So let's get out of that and let's do a little trick here. Let's do grep. Grep is a tool that will search this file, and -v says give me everything that doesn't match this mess. So I don't want to have any of the header come through, and the header's all demarcated with 2 pound signs. And when you do that, you'll get everything that doesn't have the header file in that file, and it just spits out everything that doesn't have that in it. Okay, so that's one way to search the file. Let's say we want to get every variant that is on the THC synthase config. We're going to use grep again, and I happen to know that that's config 741, and we want to search for every variant that's on config 741. Okay, what do we get? Oop, doesn't have a GZ on it anymore. Oh, it picks up the header, and then you're going to see it's going to spit out a long list of other variants that are on that large config. All right. Now, this is going to give you more variants that are just in the actual genes themselves. This is an entire multi-megabase config, so it comes out with a lot of variants. In fact, if you pipe that to word count, it will tell you how many. And give you a number for how many variants. That word count counts the number of lines. So every line is a variant in the VCF file. So this is just going to come back and tell you you've got X number of lines. Therefore, you've got, ooh, look at that, 20,000 variants on that config. It's a big config. All right, well, that's too many to look through. Let's pipe this to grep again, and let's look for only ones that are near THC synthase. And I happen to know there it's 441. Oh, this is probably, that'll get us close. That's in the ballpark. Okay, that's going to give us everything that matches 741, Then it's going to pump it into, I want everything that's got 741 and 4417. 4417 is a coordinate that's the beginning of the THC gene. This is a little bit of a sloppy search, but it will still give us a smaller list. Now it's going to search for that number anywhere in here, and we're going to get some off-target hits with 4417, but this is just a real quick tutorial on where to find these things. So, okay. Let me see. None of these coordinates are starting at the gene, so this sample doesn't have any variance there. That might not be surprising. This is in fact a reference that this probably has a knockout there, because I think this is a type 3 plant. So anyway, hopefully this is a helpful quick tutorial on how to start surfing around some of these files. Now this VCF file is important to have, because you can take this file and load this into the Koji genome browser so that you don't have to manage this terminal junk, and you can view it in a much more user-friendly graphical user interface. And we're going to touch on that in the next tutorial, is how to take your VCF file and load it into Koji so that you can begin to surf around this information in a genome browser. Okay, so that's really step one of using Canopedia, and what some of these graphs mean, and how to make the most of it. There is some other information down here. This is a QR code, which is a self-referencing QR code. If you were to scan that, it would go to this URL. That's what that QR code encodes. So you can put that on a product if you want, and anyone scans it, their phone will bring them to this page. This is some of the blockchaining certificates. So this is what you need. If we ever disappear or go out of business, your data is still circulating on blockchains, all right? And you can click on these things and get proof of your data's existence. Again, it's not putting this huge file into a blockchain. It's too big to go into a blockchain. It is putting a PDF, or I'm sorry, it's putting a hash of your data into the blockchain, which is this short signature. This signature represents your data. If you change one letter in that file, this whole hash changes. So this is such a unique number that it only can represent your file, and if you can demonstrate that this number exists in a time-stamped blockchain transaction, you can prove the date of your file. Change one letter in the file, this whole thing scrambles. So you can also prove that it's not been mucked with since the date it's been generated. All right, it's not been edited. All right, this is a kind of what we call proof of existence. We do scan NCBI for other validated data sets. Now I'm putting validated in air quotes here because these were generated with really early technologies before we had good references, and it's very rare we get perfect overlaps because there are DNA sequencing errors in these data sets. However, if we saw something that was maybe 78 or 80 out of 81 SNPs that were overlapping in the file set, that's probably a perfect match, but you might need some confirmation sequencing to confirm it. The techniques that were used here, again, predated people understanding that there's a SNP every 50 bases in cannabis, and so they use techniques that would trip up on those other variants and not always amplify targets out of the genome accurately. So we don't expect to get 100% overlap and concordance with these other techniques because they were operating with earlier tools. So, okay, with that, we are gonna go on to section 2 of this tutorial. Thanks.